Vincent Noireaux got his BSc in Applied Physics at the University of Tours, France in 1994. In 1995, he moved to Paris for graduate school at the Université Paris 11, or SAY, in physics. He did his Ph.D. at the Curie Institute, Paris, from 1996 to 2000, in the laboratory of Jacques Prost on the motion of bacterium Listeria. He studied the actin cytoskeleton mechanism involved in cell motility. He learned the biology related to this project in the laboratory of Daniel Louvard. In 2000, he joined the laboratory of Albert Lipscheber at the Rockefeller University in New York City, where he spent 5 years as a postdoc. He used cell-free expression systems to construct elementary gene networks and artificial cell systems. In 2005, he moved to the University of Minnesota, where he is pursuing his work in synthetic biology using cell-free expression. His research consists of constructing and characterizing biochemical systems by executing synthetic DNA programs in vitro, from simple regulatory elements to synthetic cell systems. Ryan Marshall, who will also be speaking, received a BA in Physics and Math from Macalester College in St. Paul, Minnesota in 2012. He joined the Noireaux Lab at the University of Minnesota as a PhD candidate in 2013, shifting into synthetic biology to work with the all-E. coli cell-free transcription-translation system. He has focused his work on genetic circuits and CRISPR technologies in TXTL. Dr. Noireaux and Mr. Marshall, over to you.
Thank you, Catherine, for the introduction. And good morning or good afternoon, everyone, and welcome to this webinar. Today we are going to discuss how to perform cell-free transcription-translation reactions on an acoustic liquid handling system by Labcyte. You see one at the bottom left. Here in the middle, you see the symbol that we use for our cell-free expression system. And finally, on the right, you see a 96 well plate with some cell-free expression reaction after a few hours of incubation. We are going to return to this extensively in the next slides. So here is the outline of the webinar today. I will cover the first two bullets, the introduction to TXTL, and then the all-E. coli TXTL system that is called myTXTL that my lab has developed. And then Ryan Marshall will cover the two next bullets about prototyping, and then Alex Pearson will present the Labcyte system. At the bottom of this slide, you have the abstract of the webinar that was also advertised on the web here. So before going into the introduction to TXTL, I would like to make some acknowledgements here. First, most of the results and the data that you are going to see today have been done by former members and current members. On the left, former members, Jong Yang-Chin was a graduate student, Mark Rustat was a graduate student, and Filippo Cascara was a postdoc. And currently, I have John Garamella, Ryan Marshall, and David Garen in the laboratory doing cell-free expression synthetic biology using the ECHO. And the work that you are going to see, again, the data have been sponsored by the following agencies that you see at the bottom. That's it. So let's start with the first section here, which is an introduction to TXTL. TX is for transcription, TL is for translation. It's a short name that means cell-free expression. That's it. So I'm going to start with a very broad introduction to synthetic biology. What you see on that slide is a definition, one of the definitions of synthetic biology and the goals of synthetic biology. And synthetic biology is essentially done in vivo and in silico, and there is a growing community doing synthetic biology in vitro, like you see on this image in test tube reactions, and using the Labcyte ECHO high throughput on well plates. That's it. So cell-free synthetic biology has, I would say, 4 major applications. The first one is prototyping. What does it mean, prototyping? It consists of testing regulatory elements or gene circuits in test tube reactions. It's an application that is popular because cell-free expression offers a system that allows a much faster turnover of testing, especially the design, build, test cycle. A second application of cell-free synthetic biology is biomanufacturing. One of the ideas is to produce very quickly in test tube reactions therapeutics, pharmaceuticals, such as antibodies. A third application of cell-free synthetic biology, it is diagnostics. You can now, for example, perform cell-free expression on paper-based substrate to screen and diagnose very quickly some processes like viruses, for example. And finally, there is a last application which is a little bit more at the fundamental level. It consists of using a cell-free expression system, encapsulate the cell-free expression system in liposomes, and build synthetic cell systems. The image that you see is by fluorescence microscopy, and the liposomes that you see are about 10 micrometers in diameter. So TXTL synthetic biology, or if you like, cell-free synthetic biology, the goal is to construct biochemical systems in test tube reactions using DNA programs. So you engineer, you clone DNA programs on the bench, and then you execute those DNA programs in test tube reaction, and using the Labcyte Echo, you can do that high throughput with well plates and plate readers. So what are the advantages of this approach? It's a bottom-up, reductionist, and constructive approach. There is no endogenous information in the system. The only DNA information that you have here is the DNA that you clone and that you add to the reaction. There is no interference in response from an organism. That has a number of advantages. There is more freedom of control and design compared to in vivo. And one of the major advantages is to accelerate synthetic biology by prototyping and biomanufacturing. So a TXTL overview, a very important point of this webinar is that there are quite a number of TXTL platforms available. On the left here, you have an example of host cells that are used to extract the transcription-translation machinery. By far, E. coli is the most popular. Most of the companies, large companies, sell TXTL systems. And in the past, I would say 10, 15 years, there have been a number of new TXTL systems based on other cells, like yeast with germ, tobacco cells, that expand the number of applications that you can do with cell-free expression. In the middle of this figure, you have a very quick summary of how a cell-free system is prepared. You grow the cell, you break the cells, you do a lysis, and what you do, you extract the transcription-translation machinery. And finally, on the right, what you do, you compose a cell-free reaction. You have an energy mix made of NTPs, of substrates, of nutrients, such as amino acid and energy source. You add the extract that provides the transcription-translation machinery, and then you can express protein and circuits. That's it. So the typical volume is from the femtoliter when you do a synthetic cell system to liters. Companies have scaled up cell-free expression to hundreds of liters. And you have a quick summary of the typical setup. You can use flask, well plates. We're going to focus on that today with the Echo and the liquid dispensing. The scale of the reaction is 1 to 2 microliters. And you can do that also in microfluidics at a much smaller volume. So on this slide, you see another overview of cell-free expression system. On the left, you have the host that has been used, where the transcription-translation machinery has been extracted, and the type of application. So the main application is to express soluble proteins, and then after you have a specific protein to express, like the proteins requiring disulfide bridges. Just a few platforms do that. You have membrane proteins, and finally you have glycoproteins. So you see that you have a summary of the capabilities of each of the platform. And again, by far, the community working or using cell-free expression uses E. coli platform. That's it. There are many platforms being developed currently from other bacteria and from mammalian cells. Just two numbers here on this slide. What is the largest DNA program that has been expressed in a TXTL reaction? It's about 100 genes. And what is the largest protein that has been expressed in a TXTL reaction? It's about a few hundred of kilodalton. I think it was about 300 to 400 kilodalton. So that gives you an idea of what a TXTL system can do. So another way to look at self-expression system is what transcription they use. And by far, 95% of the self-expression system on the market, they use the T7 promoter and the T7 RNA polymerase. It means that you have to clone your gene of interest under a T7 promoter, and then you use a kit. You see a few kits on top here to express your protein. You can use plasmids, circular DNA, or linear DNA. There have been a number of solutions to express from linear DNA. And this system, based on the T7 promoter and T7 RNA polymerase, is again by far the most popular at the level of the transcription. The translation, essentially the ribosomes, are extracted from a cell. The efficiency of those kits goes from 50 microgram per ml to 2 milligram per ml. The kinetics is about 2 to 10 hours. The temperature typically is from 29 degrees C to 37 C. And typically, you express your protein in what we call batch mode reaction, just a test tube on a well plate. Some kits have a semi-continuous reactions where you can extend the reactions using a dialysis system. On the right here, you see a typical kinetics using a T7 promoter and the T7 RNA polymerase. And here we express the reporter protein EGFP up to 2 milligram per ml. So I'm going to give a very quick overview of the type of TXTL that we have on the market. They are essentially almost all extract-based. When I say extract-based, it means that the translation machinery, the ribosomes and cofactors, they are extracted from a living cell. The cost is about $300 to $500 per ml. It's a very well-established platform, and to 2 milligram per ml of protein produced after a few hours. It is the cheapest system. The custom preparation is possible and relatively easy. There have been a number of publications reporting how to prepare those systems. You can express soluble proteins, membrane proteins, and proteins with disulfide bridges. And recently, there was a custom-made system for glycosylation published by the Jewett Lab at Northwestern. What are a little bit the negative points for this system? Is that it's limited, the post-translation modification are limited. Now you can do glycosylation, but there are a number of other post-translation modifications that are not possible, and they are listed there. On the right here, you have another prokaryote E. coli-based system. It is called the Pure system, developed in the early or at the end of the 90s by the lab of Takuya Ueda in Japan. They purified all of the components, all of the protein and ribosome that you need to do transcription and translation. It is a system that is sold by NEB. You have the cost here. It's about $1,000 per ml of reaction. It's cleaner than an extract. It's made of 4 to 5 milligram per ml of protein compared to an extract-based system, which is typically 10 milligram per ml of protein, including the RNA polymerase and the ribosomes. It has a known composition, and you can do the same as an extract-based system: soluble protein, disulfide, and membrane proteins. The downside of this system is the same as the extract: limited post-translation modification. It is less efficient, about 250 milligram per ml of protein, and the custom preparation is now possible. It's a little bit more challenging than an extract-based system. The eukaryote-based system, there are many types: rabbit, with germ, yeast, insect, CHO cells. You can see the list here. It's about $1,000 per ml of reaction. What is the advantage of the system? You can do, for some of them only, post-translation modification like glycosylation. Overall, so far, it is less efficient than E. coli. The custom preparation is more challenging. I just want to add on that slide that there is a company that is going to commercialize a new system from tobacco cells, and they claim 3 milligram per ml in batch mode based on the T7 transcription, and I gave you here the name of the company. I am not sure, I don't know if this kit can be purchased right now, but they advertise that it would come at some point in 2018. Why cell-free synthetic biology? It is fast reaction setup. Just take a Ryan will discuss this, for example, on the Echo. A 96 well plate is plated in 15 to 30 minutes. High throughput compatible, you will see that with the Echo, and very well established for prototyping. You can prototype parts in a few hours and gene circuits in just 1 or 2 days. It is safe. It is not a living organism. It cannot reproduce, does not expand. It is based on standard E. coli strains, and it's easy to use. And finally, it is quantitative. What is nice is that using calibrated fluorescence, you can quantify your expression. You can control the DNA stoichiometry, the transcription-translation component, molecular component. Their concentration is known. You can develop models, and until now, the credibility of TXTL is based on the fact that there is a very good agreement between what we see in vitro in a cell-free system and what we see in vivo. So I was saying that there are a number of kits commercially available. I'm going to describe the one that my lab has developed and what is unique about this system. It's a no E. coli transcription-translation system. What is the goal is to develop a powerful and versatile TXTL to be able to do a maximum of application in vitro. So we spent many years to develop this system, and we really focus on the reaction that you see, transcription, translation, and also how to degrade mRNA and protein to make dynamical systems. So it's a system that is not based on the T7 RNA polymerase, but on the E. coli core RNA polymerase. And doing so, we expanded the repertoire of regulatory elements to hundreds of elements. Almost everything, all the promoters, operators that you have in E. coli, you can use them to do circuits. We spend a lot of time developing the strengths of the system. We have reached 2 to 2.5 milligrams per ml of protein produced in batch mode reactions. It is dynamic. I'm going to maybe quick on this aspect. To make dynamical system, which is just one application of cell-free expression, you need to accelerate mRNA and protein degradation. And we develop methods to do this. It is scalable. We can encapsulate the system in liposomes, femtoliter volume, to milliliters reaction. And finally, we develop a system so that it's modular. We can introduce novel technologies, what we have done just recently with CRISPR, and Ryan will show you what we have done with this. So you take some cells, they are E. coli cells, you prepare a lysate, you prepare some buffers with the energy, the nutrients, amino acids, and then you compose a cell-free reaction. Here are some characteristics. The MyTXTL system, it's a cytoplasmic extract from E. coli, and again, very important, the transcription is based on the E. coli core RNA polymerase and sigma factor 70. And the translation, the molecular component of the translation are there too. We compose an energy mix and an amino acid mix to energize the system. Some other characteristics, so a TXTL reaction has a fixed volume when you do that in batch mode. What is the dilution factor? It's about 25 to 30 times compared to E. coli. You have a fixed concentration of RNA polymerase, a fixed concentration of ribosomes, and the viscosity of a reaction is relatively close to water. It's slightly more viscous, but it stays close to water. That's it. And in the table, you have a number of characteristics here with a concentration of RNA polymerase, of ribosomes. You have the transcription speed and the translation speed. This is essentially taking a factor of 25 to 30 for the concentration of polymerase and ribosomes, and you see that the transcription speed and translation speed, it's about a factor of 10 slower than in vivo. What's unique with this system is at the level of transcription. Because the transcription is based on the E. coli core RNA polymerase, we have access to many more regulatory elements, promoters, RBS, operators, and so on. You see EGFP here clone under an E. coli promoter specific to sigma 70, and after a few hours of incubation, EGFP is just produced in a test tube here. You can see here 10 microliters of reaction. And the kinetics look like this. At the bottom, you have a negative control. The closed circle, it's production of GFP using a plasmid, and the open circles, it's production of EGFP using a PCR product. And on the y-axis, you have the amount of GFP that is produced. I continue on the transcription. What is unique is that you can recapitulate the transcription scheme of E. coli. All of the sigma factors can be expressed, and you can also express RNA polymerase from phages. I'm going to take the example of sigma 28 responsible for motility. In E. coli, you express sigma 28 that is going to induce the expression from a specific promoter to express GFP, and essentially what you see is the same as a P70A promoter. You express the GFP just by doing a transcriptional activation cascade. And at the bottom of this graph, it's another negative control here, and you see the linear accumulation, the closed circle here, it's a linear accumulation of GFP. So this cell-free expression system is now commercially available. It is sold by a company named Arbor Biosciences based in Michigan. And on their website, you can look at the description of this system. They currently sell this system here. It's a one-pot. It's a one-pot myTXTL master mix. You just need to add the template DNA in the reaction to express your protein or to execute a gene circuit of interest. And I'm going to conclude that second part here with a myTXTL scope. It's a summary of some of the applications that we have done so far. You express using plasmid or linear DNA, doing transcription, translation, and we have a number of applications that you see here: synthetic biology, bioengineering, quantitative biology. We go from prototyping to self-assembly. We can entirely make phages in test tube reactions. We have a synthetic cell application, and finally, we are developing medical application with this system. And I'm going to stop here and have Ryan start the next section.
All right, thank you, Dr. Noireaux. So one principal use for TXTL is for prototyping, which helps achieve the goal of rapid characterization of DNA parts and circuit motifs. So TXTL can be used to prototype regulatory elements, and our system that we use is a bacterial system using the coli, RNA polymerase, and ribosomes, and we can prototype many regulatory elements, including the promoters, untranslated regions, ribosome binding sites, transcription terminators, RNA aptamers, transcription factors, and CRISPR elements also. And we can combine these regulatory elements to form gene circuits, upwards of 5 genes or more. And we express from either plasmid DNA or also from linear DNA, which makes it very versatile because you can just purchase a gene fragment and put it straight into TXTL to express. Typically, we use fluorophores like reporter proteins and measure their fluorescence on a plate reader to probe a circuit or an element. TXTL reactions can be done by hand, where all components are added by micropipetting. So the components you see on the top right, the list includes the lysates, an energy mix, an amino acid mix, and some other cofactors, and the DNA from which to express. So you see the example of expressing GFP from a sigma 70 promoter. So sigma 70 is abundant in our lysates, and we get the green GFP there. And by hand, there's some limitations. So there's larger error because of the pipetting. We have to pipette all of the components by hand. And also there is limited reproducibility because of that. There's no high throughput capabilities because there's a human physical limit. And I'm sure many of you have pipetted at the bench for hours, and you know that your brain and your fingers get tired. So there's that limit. It takes a lot of time. And there's a minimum volume per reaction of 12 microliters, which is relatively large compared to what we will see with the new liquid automated dispensers. Like the Echo 550 can assemble reactions with all of the same components, but there are many advantages to this system, which include it has a very small error because of the automated dispensing, which gives a higher reproducibility. There's now high throughput capabilities. So an example of a full 96 well plates assembled by the Echo. There's no longer the physical limits of the human pipetting. So we are between 5 to 10 times faster now, depending on how many reactions you're doing at once. And we've gotten the volume down to around 2 microliters per reaction, so we can save on reagents as well. So we recently put out this application note. You can find it on the Labcyte website, titled High Throughput Microliter Size Cell-Free TXTL Reactions for Synthetic Biology Applications using the Echo 550. So now I'll show a few things from that note, some data there. First, TXTL reaction volume. So this plot shows the effect of reducing the volume of reaction using the Echo 550. We see good protein synthesis for all of these cases. We're just expressing GFP here from a sigma 70 promoter. We see that the 2 microliter case is the best. And the TXTL is very sensitive to oxygenation, so the shape and the size of the droplet have an effect on the expression kinetics. And what we are looking for is the linear accumulation of the GFP, so we see that the 2 microliters gives us the best case there. So that's where we are working at right now. And we also looked at TXTL reaction setup time. So here we see a couple of plots showing that. On the left is for the first set of reactions for a single 96 well plate, and on the right is for if we're repeating that same experiment a second time. So for a well plate, we are about 5 times faster using the Echo than pipetting by hand. It takes, for one well plate, maybe around 40 minutes for the complete plate, where 15 minutes of that is the actual machine dispensing. And by hand, for a full well plate, it can take over 3.5 hours. And at that point, they're likely making errors. For repeated plates, we are even faster because the setup time is reduced. We're 8 to 10 times faster. And you can also reuse the reagent plate for subsequent plates after your first plate. Next, we'll take a look at some reaction performance. So here's an example of a gene circuit that we tested in the top left, where we have a couple of transcription cascades. So we express sigma 38, and then express sigma 28, and then finally our reporter protein as well as a repressor. And here, what we'll look at is just the comparison between the data from assembling reactions by hand versus assembling them by the Echo, which is on the top right. And they are very similar. We see the same trends. And then on the bottom, we see that we can test many more possibilities with the Echo. So this is data generated by the Echo. And on single well plates, we can see the effect of changing the concentrations of 2 or 3 different plasmids at the same time. Whereas by hand, this is less possible. So next, I'll talk about some things that we've recently done in TXTL, which is prototyping CRISPR elements. So a brief introduction. CRISPR is a revolutionary discovery for genome editing and has many other applications. It is based on an RNA-guided nuclease that binds to a target DNA sequence and either cleaves using the wild-type Cas9 protein, which is seen on the left, or it can bind to the DNA to block transcription using a catalytically dead version of the Cas9, which is seen on the right. So the Cas protein binds with a guide RNA, which contains a short spacer sequence that base pairs with a target DNA. And so far, we have successfully tested many CRISPR enzymes in TXTL. And we recently put out this year a paper in Molecular Cell that includes some of the data that I will talk about. And so all of the experiments that I will show can now be done using the Echo 550 to accelerate. So like I said, we've recently installed CRISPR in TXTL. So in a single test tube, we express a Cas protein, Cas9 protein, a guide RNA, and a GFP reporter target. And so on the bottom, we see an example of what the data might look like, where we have in the green a non-targeting guide RNA, and then in the blue we see repression when we add a targeting guide RNA. First, we characterize the Cas9 from S. pyogenes using 3 different guide RNAs targeting different locations along our reporter construct, including the promoter, UTR, and the gene body. And here are some expression kinetics for those, with Cas9 is the top row of plots and dCas9 on the bottom. And we see various fold changes in the endpoint GFP expression, and for the most part it is very efficient, maybe a little bit better for the cleaving Cas9. Next, we wanted to show how repression in cells compared to repression in TXTL to validate our TXTL system. So we used a panel of 19 sgRNAs targeting different locations along the promoter, UTR, and inside the GFP gene body on both template and non-template strands. And we also included a few non-canonical PAM sites. So on the plot at the bottom, on the vertical, we have the repression in cells, and on the horizontal, we have the GFP repression in TXTL at 16 hours. And there is a relatively good agreement between the two. And at the very least, we can see that there's a cluster of good repressors at the top right and a cluster of functional but less good repressors at the bottom left. And in general, we recover that we get good repression when we target the non-template strand or the promoter, and the repression is weaker when we target the template strand. So far, I've talked about prototyping guide RNAs when in the GFP or a promoter, but we've also developed a few ways to assess the activity of any guide RNA on a plasmid. One way, if you want to target inside a different gene of interest, you can clone it upstream of GFP and use dCas9 to bind and block transcription, and so therefore the transcription of the downstream GFP is also blocked. And so in the plot to the right, the top right, we see for 3 different fusions, we get efficient repression of our GFP by targeting in the outside gene of interest. And then also if we can target anywhere on a plasmid, not just in a coding sequence, and target with Cas9, and we cleave the plasmid, and then our lysate contains RecBCD, which is a DNA exonuclease. And if we do not add an inhibitor of that exonuclease, it will chew up the linear DNA, and we will see a reduction in the GFP expressed. And so on the bottom plot, the SG33, there we are targeting outside of our coding sequence. And when we use Cas9, we cleave and we get good repression. When we use dCas9, we don't see any repression at all because we are not cleaving and we are not blocking transcription. And so like I said earlier, we've also tested other CRISPR enzymes, and that includes the FN-CPF1, the DFN-CPF1, and E. coli cascade. So on the top, we have repression for 3 different guide RNAs for CPF1, and it works well, is maybe a little bit weaker than the S-pyogenase Cas9, so we still see efficient repression. And for the top row of plots is if we express everything from time 0, but we can also pre-express our nucleases, so our CPF1, with a guide RNA for 3 hours before adding our targets, so that we accumulate some of the ribonucleoprotein complexes before we start expressing our GFP, and we can increase the repression in that case. And on the bottom, we have similar results for the E. coli cascade, and when we pre-express, we get very high levels of repression, over 200-fold at 16 hours in TXTL. And finally, we have characterized anti-CRISPR proteins, which are very exciting because they allow for better regulation of genome editing and gene repression. So we express in a test tube reaction the Cas9, a guide RNA, an anti-CRISPR protein, and the GFP reporter in a single reaction. Then the anti-CRISPR protein binds to the Cas9 complex to prevent it from binding to the target GFP plasmid. So we tested a panel of 24 anti-CRISPR proteins against 5 different Cas9s, and we see different activities. So in total, we tested 120 different combinations, and we were able to identify ranging specificities. For example, the SPY-Cas9 and its VQR variant have very similar inhibition rates across all of the anti-CRISPR proteins, as well as the 2 type 2C enzymes that we tested, the NME and the CJE, also behave very similarly across the panel of anti-CRISPR proteins. And so overall, we can use TXTL to rapidly and scalably assess these inhibitory activities of these diverse anti-CRISPR proteins. So in conclusion, TXTL is a good way for fast prototyping of small cell-free circuits. We've accelerated the design build test cycle, and we now can characterize CRISPR elements. And so the paper that I was talking about with a lot of these data is shown here, rapid and scalable characterization of CRISPR technologies using the all E. coli TXTL system. Thank you.
Thank you, Ryan and Dr. Noireaux. As a reminder to our audience, you may ask questions at any time using the question box. Our speakers will have an opportunity to address these after our final presentation. Our final speaker today is Alex Pearson. Charles Alex Pearson has been trained as an MS in biology with an emphasis in yeast genetics. After several years in original research at Indiana University, Purdue University, Indianapolis, Alex continued as a research assistant, leading the molecular lab with the Indiana State Department of Health, and as technical consultant with Roche Life Sciences, supporting their research products and customers. Continuing with Labcyte as a field application scientist, Alex works directly with researchers to implement new workflows to meet the increasing demand for results. Alex? Hello everyone, and welcome to today's LabTools